Hello, and welcome to Chapter 15 of the AP Review video series. This is going to be regulation of gene expression, so we're going to talk about operons, as well as within a eukaryote, how do we have the different levels of regulation that come from complexities of being a eukaryote. Okay, so the first few you must know statements all have to do with operons. And so we're going to talk about inducer molecules and inhibition and repressors and regulatory proteins and all that kind of stuff. So we're going to kind of lump some of these you must know statements together. You have to understand that regulatory gene is actually a chunk of DNA that codes for a regulatory proton, part of the operon. And then you have to know the different parts of the operon that regulate gene expression in both inducible and repressible operons. So we're going to do all three of those together as we talk about operons. Okay, so we're going to start by talking about the trip operon. And so we're going to remember that operons are present in bacteria and bacteria only. And we talked about how in a bacteria, you have that one circular chromosome, and bacteria have kind of a rough gig because they're usually very subjected to environmental changes. And the example I gave you guys in class was, well, if you're a bacteria and you need tryptophan because that's an amino acid, well, the bacteria is living in your gut. For example, if you're eating tryptophan as a human, the bacteria doesn't need to make tryptophan. If, however, you are not eating tryptophan as a human, then the bacteria does need to make tryptophan. So the bacteria has to be able to respond to either of those conditions, but making tryptophan is a very energetically expensive thing to do. It takes a lot of ATP, it takes a lot of cellular resources. So you don't want to just make tryptophan all the time, just in case, so to speak. What you want to do is make tryptophan only when it's needed. And so in order to do that, we use what's called an operon. Okay, whoops. We use what's called an operon. And what an operon is a region of DNA within the molecule, the one DNA molecule, and it contains a promoter, an operator, and then all of the genes that are associated with producing tryptophan. Well, separate from this, remember we only have one molecule of DNA, so it's on the same molecule, but in a different region of that molecule, we also have what's called a promoter. We have what's called a regulatory gene, which also has its own promoter. That regulatory gene codes for what's called a repressor. Okay, so the idea behind bacteria and the operon concept is that instead of transcribing and translating all of the proteins that could possibly ever be needed and wasting a whole bunch of cellular resources, you only always code for your regulatory genes. You always express regulatory genes, but then you allow these genes to control what's happening at the rest of your genome. That's the concept behind an operon. This right here is always being expressed, but then it controls whether or not the rest of it is being expressed. So when we look at this, what we are producing from this regulatory gene is a repressor. Well, they use this kind of slightly curved shape here and then a flat shape here to indicate that this is shape recognition. I'm going to erase some of this. There is shape recognition that would or would not allow this repressor to fit inside that operon or in the operator. And if that repressor fits inside that operator, it is going to physically block the ability of the RNA polymerase to move down the chain of genes. Well, remember the RNA polymerase is what is actually required to produce the RNA polymer, and that's what transcription is. And so if your repressor is fit in this spot right here, as it's shown right there, then your RNA polymerase can't move, no RNA is made, good to go. If instead your active repressor is not present in this operator, well, the RNA polymerase can go forward and produce the RNA, and then we have translation, and we're able to make our polypeptide. So in this case, what we're looking at is tryptophan, which again is that amino acid. If tryptophan is present, then we call it the co-repressor. The co-repressor is usually what the molecule is being made. But here, if we have tryptophan present, it fits inside your repressor. The repressor has a change in conformation, And that then fits into this spot right here. Okay, that's what this is saying. And so if tryptophan is absent, then it would fall off of this. The tryptophan co-repressor would fall off the repressor. The repressor would then fall off of the operon, and then your RNA polymerase can go forward and do its thing. This makes so much sense. I know that shocks you, but it makes so much sense because tryptophan is what's being produced by this operon. So if tryptophan is present, if we have tryptophan present, there's no need for this to be occurring. So if we use tryptophan, the N molecule out here, to turn this process off, when this tryptophan molecule is used, it makes sense then at that point that the cell is going to want to go ahead and produce more tryptophan by having it fall off. So this is our trip operon. In this case, if there is no co-repressor, okay, then our regulatory protein here is not bound. If there is a co-repressor, then it attaches and then this process is turned off. So normal, okay, normal is this up here. This is telling us that this is a repressible operon because it's normally on, but it can be turned off in the presence of a repressor. A lac operon works very similarly, except it's flipped and it's backwards. Okay, it's flipped and it's backwards. So we still have a regulatory gene. That regulatory gene is always going to be on. The regulatory gene codes for a protein. The protein is active when it is first produced. In contrast to on the last slide, the repressor was inactive when it was first produced. So that's the big difference here. So here we're an active repressor right off the top, and we're blocking transcription from the get-go. Then, if the proper molecule comes and is in place, and in this case that proper molecule is lactose, comes in place, forms, attaches to your repressor molecule, that's what it's showing you down here, it falls off, and now we're able to go ahead and produce all of the proteins. Now, in this case, this is a catabolic process, or this is a catabolic goal. The goal here is to break down the molecule. And so when we talked about the lac operon in class, we talked about, well, lactose is a sugar. That sugar can be used for energy for the bacteria. And so it makes sense that if lactose is present, that then we would produce all of those enzymes that would be necessary in order to use the lactose for energy. However, if lactose is not present, it makes no sense to be producing all of these proteins that have absolutely no function in the cell if lactose isn't there. And so remember, catabolic processes are the things that break things down. So in this case, we have what's called an inducer. The inducer is the lactose. And so lactose, which needs to be broken down, binds to the repressor that causes the RNA polymerase to go through, produce these proteins. These proteins then digest the lactose. And then when all the lactose is gone, then the repressor is in its original conformation and it goes back onto the form. So in this case, the operon is normally off, okay, but it can be turned on. So that is an inducible operon. So in all of this, we also need to discuss what's called positive and negative control when it comes to gene function. So we're still dealing with the bacteria, and we're still dealing with operons. Up until now, both the inducible and the repressible operons and regulation of gene expression in that way is all called negative control. And it's negative control because you can turn it off. Okay. Positive control, on the other hand, positive control is, you can think of it like volume control, okay, on your stereo. So the stereo is on, but how loud do you want it? This is positive control. What positive control does is it uses a secondary messenger, and in this case we're using cyclic AMP, to then activate what's happening over here with the RNA polymerase. Why is this useful? Well, because here lactose is present, so we've taken that regulation molecule out of the operator and we're ready to get going. But the trick is that RNA polymerase isn't just always there. Somehow that RNA polymerase molecule has to know to go to the right place on the chromosome. And so we use active cyclic AMP, or cyclic AMP that then activates, CAP. And what happens is this is like the red flag. This is the molecule that attaches to the DNA that says, hey, 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 RNA polymerase, get over here and transcribe these genes. So just getting this guy off allows for those genes to get transcribed. The trick is that we then also have to attract our RNA polymerase to actually do that work. How do we do that? We do that here with our active versus inactive CAP molecules. Okay, so that's positive control. All right, so I alluded to this in the last video about the impact of DNA methylation and histone acetylation on gene expression. So now we're going to focus on eukaryotic cells. And when we did this in class, I told you, I want you to find 2 ways of eukaryotic gene expression for you to become really good at knowing. That way, if something comes up in an FRQ, you have 2 choices. I've never seen them ask for more than 2, but you don't have to spend too much time going through all of the different levels of regulation. So, but this diagram does actually tell you all of the different ways that we can regulate gene expression. So again, gene expression is transcription of DNA into RNA, and then translation of RNA into protein. And so in here, these are all the different ways that we can control what's actually happening within that overall theme. So we can start with chromatin modification, and that's the part about histone acetylation. Remember, histones are those little beads that the DNA is wrapped around. We can cause those beads to be very tightly packed together, or we can have them spread apart, and we'll look at that diagram in a second. And that just physically blocks or allows access of these enzymes to the DNA molecule. Once that gene is spread apart and it's available for transcription, transcription can occur. We'll talk a little bit more about transcription factors that would call to arms, kind of, the enzymes associated for transcription. We have the RNA processing step, okay, where we're dealing with removing the introns and splicing together the exons. We have the poly A tail and the 5' cap that are added to our mRNA, and we get it out of the nucleus. Once we have our messenger RNA in the cytoplasm, that's where your translation is going to happen. The control at this point oftentimes has to do with how long does that mRNA molecule get to last. All mRNA molecules are short-lived molecules, but is that mRNA molecule going to be translated once and produce a protein once, or is it going to exist for a day and be translated 100,000 times? And so either degrading the mRNA in a certain period of time is a region for control. Polypeptides themselves, once the polypeptides are created, then we have protein processing, okay, that can, we can take that polypeptide and produce the active protein, or we could degrade it at that point. So then it's up to the cell to determine how long should that protein be present in the cytosol. And then we can also then transport that protein to a particular destination. So at each one of these spots, we can control whether or not the protein is going to ultimately do its job, or if we're going to break it down before it gets to that point. Here's that histone acetylation I told you was coming. When we have unacetylated histones, they are very, very closely packed together. Versus when we add the acetyl group, as represented here by the green dots, to our histone tails, that causes them to repel each other, and because those beads then repel each other, they spread out. And so when they spread out, now we have access to all of the transcription enzymes that we would not when we were in that form earlier. So you have to know about how we time and coordinate things within your body. Well, this makes sense because, I mean, the easiest example here is puberty. When you hit puberty, whether you're a boy or a girl, all kinds of things are changing in your body all at the same time. And that's an example of a situation where all of your cells have to be on the same page, so to speak, so that they're all responding in the same way at the same time. So how is that done? Often it's done with hormones, which makes sense because when you think about puberty, you think about hormones and your hormones changing. But what's actually happening is there's some kind of, we'll go with hormones because it's an easy example, some kind of hormone floating through your body. When they find the appropriate target cell, they bind exterior And after we go through that signal transduction process, and remember the third step of that process of signal transduction is response, or the final step is response. The response is often the production of what are called transcription factors. These are molecules that now within the nucleus of your cell, there we go, within the nucleus of your cell, these transcription factors are the things that tell the cell what specific genes should be transcribed. And so that's what this is saying. We have activators, we have transcription factors, we have mediator proteins, we have the DNA bending proteins, all of these different proteins that have to come together that then say, okay, look, there's my RNA polymerase, transcription is going to go ahead and occur. And so all of these things, we have our enhancers that come in first. The enhancers bend your DNA. Your RNA polymerase then comes in once our transcription factors are there. All of these are areas that allow all of your cells in your body to respond at the same time. And the reason they all respond at the same time is because the signal that tells this to happen is coming from the outside. And it's coming from that control center, that being your brain, that's producing these hormones. And those hormones go throughout your entire body and they flood all of your cells. And then your cells all respond individually to that one similar message. And so that's how we are able to put all of this response into an organism to coordinate the response. One more you must know. You must know the role of microRNAs in control of cellular functions. So microRNAs is a part of the curriculum that didn't exist when I was in school, and it's because back when we knew that introns were being cut out of messenger RNA, we thought that introns were garbage DNA. In fact, that's what we called them. Now what we know is that some of that stuff, we call it non-coding RNA, is actually doing other things in our body. And one of those examples is MI, so microRNAs. What are the microRNAs doing? We're pretty sure they're regulating gene expression. And in doing so, here's an example of a microRNA. It's within a protein. And what this protein does is it can go along and it can find a specific sequence of messenger RNA, that's the red stuff, messenger RNA, and bind to it and literally physically prevent translation from occurring. Or it can bind to it, and then the proteins that are in this glob here can actually degrade the microRNA. So this is kind of like putting expression on hold, and this is saying, knock it off, I don't want that protein at all. What we know is the more we learn about microRNAs, the more they're in control of a lot of the gene expression that's happening in our cells. It's still very much an area of active research, trying to figure out everything that they do. So all College Board wants you to know is that the microRNAs are involved in gene expression, and here's a couple of examples why. So that's the end of this video. Thank you for watching. Take your quiz, and I hope you have a great day.